Mitosis. So in this video, I'm going to talk about the mitosis. That is the cell reproduction. So here's the three types of cell reproduction: binary fission, meiosis, mitosis. So mitosis is the most common form of cell division in somatic eukaryotic cells. When mitosis is completed, two genetically identical cells are produced. So it's a fundamental process during growth and tissue repair. All right, so here you go. Number one, interphase. The period between two consecutive mitoses is called interphase. As the cell prepare for reproduction, it duplicates its DNA and takes the necessary internal and external measures to successfully undergo the process. So cell divides into two daughter cells that can then divide again, creating a cycle of cell division. Number two, prophase. The nuclear envelope starts to break down, gradually dissolving. Chromatin undergoes progressive condensation due to supercoiling and resulting in the formation of chromosomes. The chromosome duplicates and each one moves to one of the cell's ends where microtubules are formed. Metaphase. The chromosomes align at the cell's equator during metaphase with the fibers of the mitotic spindle, and microtubules attach to each centromere of the chromosomes. Each chromosome has already duplicated during interphase, so at this point the two copies separate. Anaphase is the crucial phase of mitosis because it's when the distribution of the original genetic information occurs. The two groups of chromosomes move apart thanks to the microtubules toward opposite poles of the cell. Each set of genetic information concentrates around a centrosome. Telophase, the microtubules that aided in the migration of the two groups of chromosomes disappear, and the chromosomes start to decondense, exposing the DNA and initiating the process characteristic of an independent cell. Two new nuclear envelopes begin to form, so each containing the information of each new cell. Cytokinesis, it involves the physical separation of the cytoplasm, a characteristic ring composed of action, and the myosin microfilaments forms and constitute that cell and creating a structure resembling an hourglass. So this is the cleavage for and forms, ultimately separating the two daughter cells, each with the same number of chromosomes as the parent cell. So guys, that's all. Thank you guys and see you very soon. Keep practicing.